All right, hopefully you can hear me and see everything. So thanks for the introduction. Yeah, so the goal today is just to give a broad overview of the, let's see, it doesn't seem to be advancing, hold on. Okay, there we go, a broad overview of efforts we have in the kinase DRGC. So just a quick summary is it's composed of 6 groups. So Gary Johnson, Tim Wilson, and myself are all UNC based. Peter Sorger at Harvard University, Ben Major, who just recently moved to WashU, who previously was at UNC, along with Reed Townsend, also at WashU. And so together, these 6 groups form the Kinase Data Resource and Generating Center. And just, you've heard some, but just to clarify things a little bit, the goal is to improve our understanding of, the properties and functions of these largely unannotated or poorly described protein families. We are largely disease agnostic. So the goal isn't to focus on any one particular disease area. That being said, certainly a lot of the investigator's background, as well as the tools and systems that are really easy to study and generate data from tend to come from sort of the oncology space. So we do just sort of by default have quite a bit of a slight bias in a sense, just through where we collect of being on the oncology side. The goal is to generate and distribute these data, resources, tools, annotation of these dark kinases, with a major point being to help enable others to pursue research on these targets. So, so just to give a broad summary, there's from NIH, we basically have started off, there was a little refining at the beginning, but we have 162 kinases on our list of dark kinases. Again, actually, it's on average, cells will express well over 300 kinases at the same time, with easily close to a third of these likely being understudied kinases. So they are actually pretty well expressed and very present, and are distributed across all the subfamilies of the kinome. Only the TK group perhaps being the least well represented, but the rest are actually quite well represented as in terms of understudied kinases. So just to, one of our main goals really is to understand kinases in context of, largely in the context of their integration into functional subnetworks. So, we're really looking at trying to understand a kinase in how it relates to other well-studied kinases as well as other genes. And this is in part due to just the nature of the kinome as a whole. There is significant crosstalk and redundancy within the kinome, and which does, especially in the context of oncology, has really led to a lot of this resiliency and the capability to reprogram and bypass targeted perturbations and the creation of drug resistance. So a major approach that we have throughout is actually looking at trying to understand a given kinase by how its relationship with other kinases. So I'm just going to summarize briefly some of the major efforts we have. So one really focuses on, we are very protein centric. So there is quite a bit of proteomics work going on, and one of them is focused around the identification of local protein networks. So this is largely a bio-ID oriented process spearheaded by Ben Major, where basically we're trying to identify these local protein interaction networks around each of our individual 162 kinases. And a whole host of proteomics and sort of background sort of data integration and validation goes along with this. But the idea is to sort of, as you hopefully see on the right here, is generate local proximity networks for each of the kinases. And we think this will actually be a very good, valuable resource for trying to better understand these kinases in the context of what they're directly sort of working with or interacting with. So Another component is the identification of chemical tools. So Tim Wilson's moving this ahead. So this, really is focused on the identification of existing compounds that have appropriate properties. So for instance, potency of less than 100 nanomolar, certain selectivity and cellular target engagement assays, being done, and the creation of nanobrett assays as part of this process. So there currently are about nearly 50 nanobrett assays for our IDG targets. There's nearly 30 existing compounds that have been found that satisfy these are SGC criteria. And so this is something that's, again, being advanced in parallel with everything else. We do have efforts in phenotypic screens where this is just an example of one with basically a dye drop assay or basically the ability, this is Peter Sorger slide from him, where trying to look at the basically labeling of different cellular components and trying to do sort of large scale phenotyping of cells in response to various perturbations. So there's other phenotype screens that are also part of this process, but we have a number of these. And another is, again, sort of quantitative Kinome Dynamics. So this is a pretty large effort that integrates multiple groups, including Reed Townsend and Gary Johnson and myself and others actually. So where here the main idea is we do some types of cellular perturbation that could be CRISPR or drug treatments. We're doing a lot with drug treatments right now. And it's utilizing the SMIBs, multiplex inhibitor beads, where we have covalently linked inhibitors to cephalose beads, and cell lysates are washed across these columns. The kinases bind, and we wash those out and run them through mass spec, and basically quantify the kinome in mass. This is done in parallel with actually PRM, parallel reaction monitoring, to do quantitative proteomics of our kinases of interest. So Reed Townsend has already basically created kinase-specific peptides, PRM peptides, for each of our 162 kinases. At least, I believe that we have at least 2 for each of them. And so this allows us to quantify at femtomolar levels the amount of kinase in a sample. So this is these perturbations combined with quantitative proteomics is being used to basically really probe and alter the system, and then measure readouts from that, their response, whether it's at a transcriptional level, a proteomic level, and others. And then this feeds into obviously a lot of integrative analysis to try and understand, again, our understudied kinase in the context of other well-studied ones. So this is all integrated into, we're putting together, we have a website, darkkinome.org, that helps to integrate and distribute the data that we have. So even the raw data, GitHub repositories for code, everything is sort of integrated into this. And then along with Pharos, which you heard about yesterday, we work to try and share data that is appropriate for both sides so that we can get it out to as many people as possible. So we also have associated tools to help work with both our data as well as others. So just as an example, Indra is essentially an NLP system that grabs data about relationships between, and we're using it specifically for our understudied kinases and all the relationships sort of surrounding them. Those could be protein interactions, but those could be transcriptional regulation, drug information, anything of that sort. And these are integrated into networks for each of our kinases. We have expression browser that has a very kinome-centric view of the expression of a kinase in a given tissue relative to other kinases. So you can, again, much more focused on the contextual understanding of a given kinase. So again, a major focus of ours is to develop tools that help enhance understanding and further analysis of both our data as well as integrating with other groups' data. As an example of the scope of at least tools that have currently been identified for these kinases. So the ones that are purple have both, or have just the compound. If they're green, or if they're orange, they have the nanobread assay, and if they're green, we actually have both for them. So just to give a little bit more detail and example, focusing on cancer and understudied kinases, we used MIB-MS. Again, this is work with Gary Johnson, where we explored, basically looked at different subtypes of breast cancer. So Claudin-low and basal-like are classically triple-negative breast cancer, and then HER2 and luminal are their own subtypes. And again, lysine samples, isolating the kinases, you can sort of see on the right that we can capture nearly 90% of the kinome through this approach. So it's a very powerful approach from that standpoint, and a vast number of understudied kinases are captured as part of this process. And so we found that, for instance, we can look at just even if we treat these kinome responses as profiles, the same way a gene expression signature, this would be a kinome protein signature in that sense. That on the right, we basically have columns of samples, and each row is a kinase, and you can see that the red and blue cluster together. This is showing that the profile of the triple negative, the basal and Claudin-low, are similar enough to each other, distinctive enough that they actually, you can classify them as a group. Same with the pink being HER2, they all have similar kinome profiles, as well as the luminal. And again, simple things like PCA and stuff show very distinct groupings of these, indicating that really that this MIB binding profile actually has quite a bit of information within it. So we've looked at this further, trying to identify what's the most informative kinases in these breast cancer cell lines, at least. And so we identified, just through some feature selection techniques, 50 pan and subtype specific kinases. So kinases that are either very predictive of a given subtype of cancer, or B2, for instance, is highly predictive of it being a HER2 enriched subtype, obviously. While other kinases could show perhaps graded levels of not expression, but presence on a signature in these kinobead or MIB-MS profiles, such that you can distinguish different subtypes. And so when we find the most informative features from this, we found it to be highly predictive. What was also interesting is nearly half, a solid third, would be these understudied kinases. So what we're finding, right, is that there's a large number of understudied kinases that really have very distinctive profiles in each of these cancer types, which really suggests that they are potentially valuable targets and understudied targets for future therapies, perhaps. So another thing that we found is that this was all based on cell line data, but we have found that it is predictive of patient tumor biopsies. So this was needle biopsies from patients. So we had 2 HER2 positive and 5 triple negative biopsies, and the support vector machine was developed to just try and classify, learn from the cell line data to try to make predictions of the biopsy. And basically it was able, HER2 just fine, and 4 out of the 5 of the triple negative, it was able to make accurate classifications on these profiles. So that was quite encouraging, in that even the cell line data has predictive information about patient samples, especially since a lot of the patient samples don't have full coverage of all the kinases that we typically measure. So that's been linked then also with perturbation, because we're really interested in understanding how the kinome responds to perturbation. And so just highlighted here in this blue box, we have basically SUM159 and HCC 1806s. Both are in this triple negative, but one's Claudin-low and one's basal. And so given the same treatment, of 100 nanomolar trametinib, they actually have very different responses in terms of the kinome, how they respond. And they're both technically triple negative, but they are, one's claudin low and one's basal, and they actually do have very distinct responses. So in the process of looking at this, we can again see on the left here, the green highlights specific understudied kinases that have very strong responses and to drug perturbation, again, indicating that some of these could have be very interesting to follow up with in terms of their role in regulating signaling in response to drug treatment. We've also looked at more the subnetwork level to identify differences, and again, subnetworks of kinases within the kinome show, again, different distinctive behaviors. So just to highlight some other, this tree on the lower left is just an example, and I believe SUM159 cells of the trametinib response. So you'll see the really very dynamic, in some cases, even larger, very large responses across the kinome to a given drug treatment. And again, understudied targets are very often a major component of this. There's a number of other targets that we've identified so far. So we've done CRISPR studies where we've identified a set of kinases that really seem essential in various cancer cell lines, metastasis, homeostasis in certain cases. So we've already started to identify a fair number of potential targets for further lead development or target development. So, and right now we're really expanding this quite a bit. I guess I kind of showed this before, but again, further, more detailed. That previous study didn't involve PRM, and this, we have a new one that involves PRM, broader protein perturbations with therapeutic compounds, and again, quantification of response. One last thing is, this sort of came up yesterday, so I thought I'd just throw it in, that it's very much related actually to the embedding that was in the previous talk, is we are looking at, again, the structure of this little circle here of showing the kinome in different subnetworks within the kinome, identified through different physical interaction data and other types. And again, the green show that understudied kinases are distributed throughout the kinome, but in distinct subnetwork compartments. And we're sort of using that as another way to, rather than look at the response of an individual kinase, we're looking at the response of sets of kinases to perturbation, and then looking at the output response, so whether, to a given drug at different doses. And one of the things we've found so far, right, so on the right here is again, is an embedding, like what was discussed in the earlier talk, where each point represents the kinome profile in response to a drug perturbation. And the color represents the phenotype of that cell. So in the middle, there's sort of a cluster you can see of yellow, which means that despite the drug treatment, the cell really didn't care. When it turns green, it actually seemed to give a cytostatic response. And in the darker, it represents cell death. So you can imagine, again, if you can, at least the theory, right, is if you could I'd move the kinome, the state of the kinome, into a particular region. The prediction would be is that you would get that sort of phenotypic response. So that's something that we're trying to follow up on and trying to better understand how we can link perturbations through the kinome to some downstream phenotype. And again, what the role of understudied kinases is in providing that response. So
Shawn, can you wrap up? Oh, good.
I'm done right now. So thank you. And those are, a lot of people are involved in this, so I'll take any questions whenever. All right, thanks.
Thank you for a very